Good morning. Nice to see you all here today. My name is Leanne Stewart and I'm the Chief Executive of Kiwifruit Vine Health. And a very warm welcome to the 2021 Biosecurity Day on behalf of KVH and Zespri Innovation. So we're particularly happy to see you today here in person, given the COVID challenges in other parts of the country. Today we've got a very exciting program for you about research that's been undertaken over the last year and upcoming priorities. So the research program plays a very important part in answering the questions we have about enhancing the prevention of keeping exotic pests and diseases out of New Zealand, as well as preparing for them if they arrive, so having tools in our toolkit. So today's a great opportunity for our speakers to share some of the research outcomes with you and how we hope they'll be practically applied in the future. So today we'll be hearing from Matt Dick on KVH's SWOT analysis. David Chulon will provide us an update on B3 activities. Brendan Gould will share with us his experiences on forestry biosecurity, and then we'll have an update from the Zespri KVH R&D projects from Kerry Everett and Monica Walter from Plant and Food and Shandon Powell from Zespri Innovation. We'll then let you have some morning tea and then we'll come back and we'll hear about fruit fly and BMSB. So Matt will provide us an overview of the GIA Fruit Fly Council. Nicola Robertson will share an overview of the GIA BMSB Council, and then our last sessions for the day will be on BMSB long-term management. So we'll hear from Shane Max of Zespri, Matt and Yannicka from KVH, Osamu Francis from Zespri, and then we'll have who will take us through an interactive session, and then we'll have a panel discussion with Shane, Nicola and Mike Montgomery, a kiwifruit grower. We'll wrap up the day with Juliet Ansell from Zespri Innovation, and then we've got some lunch available for you before we go. So our first presentation this morning is Matt Dick, our Biosecurity Manager from KVH. Originally, Matt has a background in marine science, but he saw the light 8 years ago and joined KVH. So he has a wealth of experience in biosecurity risk management and the kiwifruit industry. So welcome, Matt.
Okay, good morning everyone. It's not that unusual to come from marine science to biosecurity. There's a few of us in the room today, so good background to have. Okay, so this was going to be a joint readiness presentation with MPI to showcase some of the work that we're doing under the GIA partnership. Unfortunately, as a result of COVID travel restrictions, they couldn't be here today and send their apologies. It also allows me a bit of deviation to address a few things that I wanted to. So GIA, essentially, for anyone who doesn't know, it's our partnership with both the Crown and other signatories to share decision making and costs of biosecurity readiness and response. But where I thought I might start is we've got a number of KiwiNet members here today. KiwiNet is our group of biosecurity champions across the kiwifruit industry, mostly post-harvest, but extended out broader as well. And we meet twice a year just to make sure that if we have a response, we're all up to speed. At our last KiwiNet presentation, we had our meeting in July. We had a really good presentation by Mike Chapman, who challenged us on how well we've learnt or how well we've responded to some of the lessons from the PSA response. So I thought I'd just quickly start there. Basically, at the end of the PSA response, or in about 2013, we had a review done by the Superior Group, which identified a number of recommendations. Now, this middle one here was one I thought would be a good one to address, and that's really that kiwifruit was operating in a bit of a silo when it came to biosecurity readiness and even in the response. There was a number of learnings that we could have taken from other industries, but also from the Crown. And I thought this would be a good place to start because it relates really closely to GIA. So GIA, we have 22 partners representing 24 sectors that come together to talk about biosecurity. This is representing about 80% of New Zealand's primary industries, and it's the single largest gathering of all industries coming together. We have animals, aquaculture, horticulture and forestry all coming together and talking about biosecurity. So that's a really powerful thing. And we have the likes of Brendan Gould and Nicola Robertson, other biosecurity managers here today, to make sure that we're sharing those learnings and working from each other. And of course there's MPI as well, who are represented in this partnership, and they have a wealth of biosecurity expertise. Now, one of the lessons of the PSA response is that we could have tapped into that perhaps a bit better, and it's certainly something that we're working on at the moment. As an outsider, you might think that biosecurity responses are quite a rare thing, but if you've worked at MPI, you'll know that the response team is one of the busiest teams in the organisation. There's always a response going on, and actually at any given time there's typically about 20. So as of March, there was 24 active responses going on. And one of the reasons why this is quite significant is where you don't eradicate a pest, you end up with another pest to manage ongoing, such as PSA. So your pest burden gradually increases year upon year. Now for growers, that's significant, but also for biosecurity risk, this is significant because this is a situation that's effectively happening in pretty much every country in the world. The number of pests is increasing, and this means the number of pathways that we have to protect is increasing as well. Australia have done a number of good studies recently, and this shows this graph here. Your light blue down the bottom is your incursions, and then the dark blue on top is showing the increasing pest burden of these responses that aren't eradicated and still have to be dealt with year on year. What this means for us, this is our closest neighbour, it's probably the biggest risk given how connected we are, means that effectively biosecurity risk from a pest perspective alone is increasing, but then this is further exacerbated with the interconnectedness of all our countries. What this means is that we can't keep doing the same thing. We can't just scale up our current approach. We have to do things smarter, and there's a bit of a delay, but there's a statistic that actually, even if we triple our investment in biosecurity, we won't keep pace with this increasing risk over the next 10 years. So we have to do things smarter, and sharing with other sectors is a key part of that. We can't afford to work in silos, and it's also great to have people like Dave Toulon here talking about some of the work that B3 is doing, which is really around that smarter technology. So that comes to some of the work that we're doing in partnership with MPI. So we have a number of initiatives that are specific to kiwifruit specific threats. Kind of group them in these categories: diagnostics, making sure that if something arises, we have the tools to detect it, having readiness plans in place, testing those readiness plans with simulations, and then also doing baseline surveys to understand what's actually out there in our industry. This is also done in partnership, of course, with Zespri Innovation, and we're hugely supportive of the contributions they provide us. So some of our current work that we're doing is shown here, and in particular we've been working a lot on Ceratocystis, the soil-borne pathogen that's in Brazil, and some of that work's been revolved not only around diagnostics, but making sure we understand which strains are pathogenic to kiwifruit, and then how tolerant or susceptible some of our kiwifruit cultivars are. If we think back to PSA, that was a real key tool in the toolbox. So we're trying to understand that for some of these other threats as well. Now that's some of the work that we're doing for kiwifruit specific work. And then under GIA, we have this other, what we call multi-sector approaches as well. Fruit fly and BMSB, which we'll hear of later today, but also things like Xylella. Now Xylella is an insect-vectored bacteria, which is a significant threat, probably the number one threat to the wine industry. It's also decimating the olive industry throughout Europe. There's no evidence to suggest that kiwifruit is susceptible to this, But given the impacts to some other industries, we can't afford to not be in the conversation. So we're just making sure that that readiness work is progressing and that we're involved in the conversation. The other one is Lepidoptera, which is basically your butterflies and moths. And by having these broad categories now so we've got soil-borne pathogens, we've got our fruit flies, our sap seed, our insect like BMSB that feed off the nectar of the fruit, your insect vectored and your insect and moths. We have these really broad categories for our readiness work, which means that if it wasn't this specific pathogen but something else, we've got a general idea of how we might respond. Obviously working a lot with the kiwifruit industry as well. So Zespri innovation, as we mentioned before, we'll hear a lot about that today, the work we're doing together. KiwiNet, this is a really key strength for our kiwifruit industry, and of course the work we're doing with growers. That on-orchard biosecurity is really fundamental. Now we talk about, there's the diagram of biosecurity not being a wall at the border, but a net that reduces risk as you go through these layers. Well, the final layer is your orchard boundary. That's where you mop up any final risk. It's really important. So it brings me to where the gaps are. And what I thought when I was thinking about this is there's gaps, I think there's three main gaps that we can work on, and some of these we can address both through the proposed pathway plan, which is coming into effect from 1 April next year, but also through innovation solutions. So one of these, which was outlined in the Superior report, is that we can't assume that organisms will be detected on day one. There's a lag phase from when something arrives to when symptoms are expressed. We see that with the likes of COVID, that's a period of days, but for some of these plant pathogens, it could be months or even years. The earlier we can detect an organism, the more chance we have at eradicating. And of course, as I mentioned before, if we don't eradicate, we get this cumulative burden. So one of our big gaps is how do we detect pathogens early? Now, growers reporting the unusual is a really important part of this. Linda's done a lot of work in this space in particular, but encouraging growers, do your monitoring. If you see something unusual, let us know, and we provide what's normally a free service to figure out what's going on. This is a key part of our surveillance, but we realise that if the symptoms aren't obvious, then it does have its imperfections. So the innovation solution here is what does surveillance best practice look like for the kiwifruit industry? And as a innovation portfolio, we're certainly considering projects that we could do in this space. This is a key part of the pathway plan as well. Monitoring, looking for anything unusual, and not providing plant material that's symptomatic. The other is traceability. Again, a key part of the pathway plan. If you're providing plant material, keeping accurate records of who you're providing it to and where it's gone. And if you're sourcing plant material, knowing where you've got this material from and where it ends up in the orchard. This gives us the ability to trace if anything unusual starts popping up, and we can see where that might have come from or where it may have gone to. That's really important. Key part of the pathway plan. But as we've seen, that if there is an incursion, wading through that data, if it's all on paper, can be a very time-consuming and slow process. And if we come across records that are inaccurate, that creates complications. So there's a real opportunity here for innovation to deliver some solutions as well. And that's something that we're working on to develop better tools for traceability. And the final one is it's kind of biosecurity complacency, but I appreciate that could be a bit rough because it's not just complacency. There's other factors involved as well, such as compliance overload. There's just becoming a grower can appreciate there's just more and more things to do that there wasn't in the past. And sometimes biosecurity, we hear that yes, biosecurity is really important, but I've just got a task list that's really long and it doesn't sit at the top. So what we've been focusing on as part of the pathway plan is getting out there running workshops with growers. We did our first one in Nelson yesterday. How do we complete biosecurity plans? And then the innovation solution, which Lisa's been working on a lot, is having these online tools so you can complete your biosecurity plan online and come back to it and keep it up to date. So we'll be running Bay of Plenty workshops in early December, one in Te Puke, and we'll have details on that in today's bulletin coming out. So I think that gives us a bit of an overview, and yeah, I hope to see you at our biosecurity workshops. Thanks a lot.
Okay. Thanks, Matt. We do have some time for a few questions if you'd like to stay up the front. So does anyone have any questions for Matt on his presentation?
Thanks. Matt, I was curious about your comments up the front of the presentation around a lesson learned out of the PSA inclusion was perhaps we could have got more out of interacting with the MPI inclusion group. I sort of remember them having quite a presence, so I'd be interested to know what you feel could have been explored further.
Thanks, Sonia. So good question. Of course, I wasn't here in the early days of the PSA response. I joined in 2013. But effectively, what the report was saying, where this recommendation came from, is that there was almost two responses running, and whether that's fair or not, but there was a Wellington-led response and a local response as well. And sometimes those weren't that well connected. Now, it may be that we were operating a bit in a silo, but it may also be that MPI was operating a bit in a silo from Wellington as well, and there just wasn't that connectivity that you'd hope to see. I don't know, do you think that was fair? You were here.
I guess you're sort of triggering a little bit for me, but probably reflecting on the recent P-Weevil incursion, because I've heard from people that were involved in that response that there was a very Wellington versus local kind of setup. So perhaps that is definitely something to follow up, but definitely in partnership with MPI.
That's right, and I guess in some fruit fly responses we've done things like I've actually flown to Wellington and been in the response team so that when industry have concerns we have a person within the organisation who can help engage with the team down there.
Just not in defence of MPI, but just providing some information, both the PSA response and the P-Weevil was prior to GIA being in place. So it wasn't under that formal partnership of cost sharing and shared decision making. So hopefully a lot of that disconnection has been addressed now that we are in the partnership framework. Yeah.
Sorry. I was just asking about Zyala, Matt. How great a risk does it pose to New Zealand as an incursion?
Sure, so I'm certainly not the most informed person in the room to talk about this, and people like Dave have done a lot of work. But there's two components to Zyala. You need the insect vector to spread it around, and you need the pathogen. Now, we've got some work to understand what vectors we may have in New Zealand and where those may be present, but certainly it's high on a number of industries' radars as a potential threat, would be fair to say. David, I mean, you've done a lot of work in this space. Any comments on that? Is that fair?
I've managed a lot of work in this space.
Yeah.
Yeah, I mean, there is one vector in New Zealand. Sorry. There is one vector in New Zealand that's the problem one in Europe, but there are other ones that we don't have.
Yeah.
Yeah.
Okay, thank you.
Great. Thanks, Matt. Some really good key messages there about the importance of reflecting on lessons learned from a response that did have a significant impact on the industry. Making sure there's continued investment in biosecurity, excuse me, and that importance of collaboration as well, so readiness in the kiwifruit industry and then across the GIA partnership. And as Matt mentioned, we will be having some on-orchard workshops coming up in the early December, so keep an eye out for those. So next up we have Dr. David Chulon, who's the current Director of Better Border Biosecurity, or otherwise known as B3. B3 is a national research collaboration with partners in research, government, industry, communities and Māori. David has postgraduate degrees in horticulture and entomology and has worked in research institutions and universities in the Netherlands, USA and Germany. David has broad experience in plant protection, plant biosecurity and in biodiversity in a range of plant systems, specialising in biology and management of small insect pests. So welcome, David.
Thanks very much, Leanne, and thanks for the opportunity to talk to you today. Yeah, I always get a bit scared when people ask me about specific things on research, because I sort of dabble on the edges. So this has a delay on it, I believe. So really, this is just a graphic of B3. As Leanne's pointed out, it's a partnership of research organisations, government and industry. It links to the Kiwifruit industry through the Biosecurity Steering Group and also the Plant Biosecurity Council. And Leanne is on the Collaboration Council of B3, so we have good links there as well. For those of you that follow the science politics that are going on at the moment, B3 is a little bit like what appears that the government wants in the green paper that's just been released. It targets an issue of national importance and it vests and collaborates across a number of the research providers. And that is what the government seems to be looking at the moment. So this schematic gives a bit of an example. I've got to keep an eye on this time thing over here, of what B3 does. So our framework, our research framework, our current challenges, and our ways of working. And this is designed to minimise the entry and establishment of invasive species that threaten New Zealand's valued flora. So we work in plant systems, productive sector systems, and natural systems, and the interactions between those. And there are some of the outcomes that we will be targeting through our research. I also put this slide up here, and Matt has touched on this. This is the biosecurity continuum from on your left, the very beginning of an invasion to the right, yes, that's right, to the long-term management. And you can see there on the top bar that the research return on investment is much higher if you can keep the thing out of the country, and you must know that. It's pretty bloody obvious. B3 works very much at the left-hand end of that continuum, so we're there to provide science solutions that stop things coming into the country. And these is some of the science that we do. So we've divided B3 into 5 themes, and the sort of questions we ask within those themes: what are the biocontrol agents of risk to New Zealand, and should we work out how not to bring those in? What are the high-risk pathogens and pests to New Zealand? What are the pathways that they might come in on, and how do we manage those pathways? If they make it here, can we identify them as quickly as possible to know if they're going to be a problem or not? And can we pick them up through surveillance early? Because if we know we get them early enough, we have a much better chance of eradication. So that's the sort of questions that we ask in our research. Now, I'm going to provide some answers, some examples from each of those themes very briefly to illustrate the sort of research that we do. So theme A, this is risk assessments for international introductions. Now, all of you are very aware, I'm sure, of the preemptive biocontrol program for BMSB, where Samurai Wasp went through the EPA program and was given permission to bring it into the country under certain conditions. This project is a new project that we're looking at the generalisations around that. So the next time that comes along, and it might be the spotted lanternfly or something like that, we have all the understandings of how we can do that quickly, etc., etc. So this is it's only a 1-year project. It may extend into a 2-year project, and it's working with international colleagues in Fresco who see this preemptive approach to biocontrol as being something that is very, very useful and should be considered at the international scale. So that's theme A. That project's been led by Gonzalo Avila in plant food research with colleagues from other CRIs. The theme B, this is risk assessment for unintentional introductions. So this is the sort of normal thing you think about trying to stop things coming into the country for pests and disease. B3 has led the need to access Chinese literature in this area. China is considered one of the central nodes for the spread of invasive species. It's also our number one trading partner and was our number 2 source of tourists, so quite important to us. There is a lot of information we've found in Chinese language and in Chinese databases that is not normally available to the biosecurity community. And of course, it's especially important for kiwifruit because kiwifruit comes from China, so there are a lot of pests and diseases sitting in China that we don't know a hell of a lot about. Zespri, KVH has been funding some of this work specifically for BMSB and spotted lanternfly, but also other pests and plant pathogens. How am I going for time there? Not too bad. So that's theme B. Theme C. This is our pathway risk management theme. And my computer's going slow as well. There must be something about this room. So the idea of the research in this theme is to detect species in the pathway and manage them on that pathway. Here we have an example of detecting of a technology that's being developed to detect fruit fly, and this would probably be the sort of thing that would be used in a transitional facility, for example, before these things were released. So this is a novel sniffing approach using the extremely sensitive olfactory receptors of insects. And again, it's taking those, developing them to a sensing panel, applying algorithms, and then being able to detect fruit fly in those situations. This project, unfortunately, has been severely affected by COVID, but it is still one of those projects of high risk, I think, but certainly huge potential. Right, moving on to theme D. So we have a diagnostics theme. And this is an example of a project that Marion Wood's developing from Plant and Food. So it's looking at in-field diagnosis to support the lab testing that is normally undertaken in a response. Again, this is quite novel. It's using DNA, nucleic acids, to develop a miniaturized electrochemical circuit that will ping when the disease is being detected. This is proof of concept, and we're looking at the next stage as to how this could be uptaken within field surveys. And finally, theme E, surveillance and eradication. This is a new project. Sorry, my computer's not keeping up with me here. So this is another new project that's just been initiated within B3. It's being led by Federico Tomasetti from Ag Research. And it's looking at remote sensing, potentially using satellites, planes, or drones. The expectation is that this sort of technology will provide significant efficacies in responses. And it will be building off international develops in this area. One of the targets for the research here is looking at where the tree of heaven can be found. So if there's a BMSB response or even a spotted lanternfly response, it's going to be very important to know where the tree of heaven is because it's one of the significant pests of those, it's significant hosts of those pests. So we'll need to know almost where every last one of those is. And it's hoped that remote sensing will actually help that significantly. So those are some of the examples of some of the research that we're doing in B3. There's 30 odd projects there at the moment, so that's 5. Can't go through the other 25, of course. So my last slide now, and again, Matt alluded to this slide. New Zealand's biosecurity system is made up of a series of interventions across the biosecurity continuum. And B3 works closely with government, industry and various communities, including iwi, to provide science solutions at various points in that continuum. Thank you. That's me. Do I have to answer questions?
Yes, thank you, David. It's great to hear about B3 and for you to share this information with the team today. B3 is a very successful collaboration and partnership, and I think that other disciplines could learn a lot from it in the research area. The themes do closely align with the KVH Zespri biosecurity portfolio, and the projects both past and present are highly relevant to getting tools in our toolbox. We do have a couple of minutes for some questions for David, if anyone would like to ask some.
Hi, I'm Juliet from Zespri. You might not be able to answer this because it's just a bit of a technical question. I was really interested in this tree of heaven because I'd always understood that BMSB are pretty not fussy about where they feed. So why is that one of particular interest?
So BMSB is not that fussy. My understanding, though, that it's less fussy about Tree of Heaven, so that is one of the plants it goes to. I think the other thing about Tree of Heaven also is it's a weed, it's a noxious weed, so it's growing in various positions around the place, including in urban environments. So it's possibly not something that a grower would be looking at, for example. Okay. Yeah, it's actually a noxious weed, I believe, but not being controlled. But my understanding it is, and we've got some BMSB people in the room, it is one of its preferred hosts. So it is one of the places you will actually see it early on in the invasion cycle.
Okay, thank you.
David, I have a question. Leanne emphasised, and I do want to re-emphasise, that I think B3 is one of the success stories of research entities collaborating, and I know how hard it is. Can you comment on your B3 collaboration with offshore research entities? Is there much of that as well?
Yeah, there is. There's quite a bit, and I only had 10 minutes today. So we work, we have MOUs with 2 Australian partners, so the Plant Biosecurity Research Initiative and CIBRA, which is another research entity in Australia. We work with UFresco, which is an international organisation through MPI. We work, and these are the high-level interactions we have. Who else do we work with? I'm going along to the International Bioeconomy Plant Health Working Group. That's another entity. And we have 2 of the partners have MOUs with one of the major institutes in China, et cetera, et cetera. And you will find just about all of the programs have their own collaborators, international collaborators, all the projects. And we often don't point that out. In fact, in the annual report, I pointed out that something like 80% of the publications that are coming out of B3 are co-authored by international researchers. So we have a very heavy interaction with the international science community.
Hi, David. You talked about insects. You mentioned, I think, one pathogen, and you really didn't talk about weeds.
Yeah.
How is the funding proportioned, and do weeds really matter to us? Up here, so many weeds. I'm so amazed.
Yeah.
I'm new to New Zealand.
So Monica, I didn't actually. I should have realised you were sitting in the audience as a plant pathologist. So 3 of the examples were on insects, and 2 were on pathogens. Weeds is a growing area. It's possibly not a good example for kiwifruit. I'm not sure about that, but we do certainly have a number of weed projects within B3. And weeds certainly are an issue, yeah.
Great. Thank you very much, David.
Thank you.
So next up we have Brendan Gould from the Forest Owners Association, who's the biosecurity manager and started at the beginning of 2020, right when COVID emerged in China. So on Brendan's first day, he experienced the implications of a significant biosecurity issue on trade and the supply chain when logs were stuck on the wharves in China. He's responsible for leading and working closely with industry participants to improve biosecurity in forestry. Prior to working at FOA, Brendan had many roles in MPI, his last role being the Manager of Biosecurity New Zealand's Surveillance and Investigation and Incursion Group. So today Brendan's going to give us some insight into the plantation forestry sector and its approach to biosecurity risk management. Thanks, Brendan.
Thanks, Leanne. And just heads up, I'm one of those marine scientists as well. Look, I thought I would start off just a few facts around the industry to give you some quick insights. So 2.1 million hectares of plantation forest, of which about 1.7 are productive forest, standing value of over $50 billion. About 33 million cubic metres of wood was harvested last year, 20,000 jobs, roughly, give or take. And then in 2020, the industry was valued, or its export earnings were valued at about 5.65 billion, and this year, earlier in the year, projected to be about 6.25. It's been a pretty good year for forestry, so I suspect we might have surpassed that. 87 to 90% of the industry is based on a single species, so radiata, Pinus radiata. Douglas fir is the next most popular at 7%, and I think natives make up about 0.3 or 0.03% of forestry. And forestry is a long game. So trees are in the ground for 15 to 40, 50, 60 years. So high expectations on forestry and the contribution that it can make to, I guess, New Zealand's future, particularly as we transition to a lower emissions future, whether it's through maximising value to New Zealand through the industry transformation plan, our contribution to a circular bioeconomy, sustainable wood-first building, but also through the many and very significant non-market value contributions that forestry provides, whether it's mountain biking, erosion control, water quality, biodiversity, recreation. There's a whole range of other values there as well, which we don't necessarily benefit from as an industry, but the public do. And of course, there's carbon sequestration as well. Sorry, we'll slowly catch up. So the Climate Change Commission has signalled an expansion in forestry is required if we're going to meet our climate goals. And why is biosecurity important to forestry? Sorry, we'll get there. Well, primarily to protect forests and forest health. I guess that's our interest as an industry. And as I said, the trees are in the ground and exposed to biosecurity threats for a very long time. So that's a lot of nail biting for investors. So investor confidence is also something it protects. Protecting market access, and then those non-market values I talked about, which have been estimated to be 2 to 4 times the actual market value of productive forest, and in some areas up to 10 times. So, it's a significant value there. And just to give you a quick insight into the structure of biosecurity, so we have a long history and a proactive history in the biosecurity space, dating back to when forestry first started here. There are 2 main grower representative bodies, the Forest Owners Association, so they're the big corporates, and then the Farm Forestry Association, and they're kind of the smaller growers, smaller investors. And then we also have the Forest Nursery Growers Association. I've put them down the bottom. They're not a levy payer per se, but they are an important part of the sector. And then there's the Forest Biosecurity Committee, and that oversees, it's a pan-industry committee with representation from all of those groups, MPI, Te Uru Rākau and Scion as well. And they oversee all the, I guess, the forest industry's relationships and interactions and the delivery of forest biosecurity across the sector. And you can't quite see it there, but it's a pretty messy diagram. There's a lot of relationships and there's a fair bit that's going on. And this is all funded through the Harvested Wood Levy as well. So at the very top there, we've got the Forest Growers Levy Trust. So they oversee and prioritise and give us funding to do the work that we do. And the levy, I guess, funds a whole range of industry good activities, of which biosecurity is just one. Research is probably the biggest beneficiary of the levy, at over 50% of what we get. We're about third in the list, so we do pretty well, and it's actually seen as a huge priority for industry, and a lot of our research is funded or contributes to research for biosecurity as well. So our pot is actually slightly bigger than that as well when we dip into the research that we get. And we also have a biosecurity response levy, as you do, that's set at zero, because we touch wood, there isn't any in here, to fund any responses that we have. Now, I thought what I would do is just to give you a bit of an insight into biosecurity, maybe. We'll see if we can get there. There we are. I might just talk through the 5 R's. So there's really 4 R's and then research on the end. And that's basically the biosecurity system, if a different way of looking at it. But before I do that, I guess a very quick flavour of where biosecurity is in the forestry industry. It's in a pretty good place. And we kind of tick most of the boxes across the system, but there are a few areas where we've got some gaps and a lot of improvement to do. And the industry has developed a biosecurity strategy to try and guide how we might improve on some of those amber areas, and also do a little bit of enhancement in some of the slightly off-white beige areas as well. And that's, I guess, a 10-year plan to help us with our direction. And these are pretty loud, I apologise. I'm just going to whip through these pretty quickly. So reduction, the sector's actually done a lot of work in this space over the years with MPI, predating GIA. And the measures that are in place are reasonably effective. There have been some reviews of them as well. There are gaps, as there are across any sectors. But we're kind of fortunate and unfortunate. We don't have an import health standard that allows us to bring in forestry germplasm. That was canned a decade or so ago, possibly longer, when pitch canker was detected in some imported Douglas fir. The industry decided it was probably safer just to not import stuff. So that's kind of blocked up a fairly significant risk pathway for us, but it also limits our ability to import new germplasm. But there's a lot of other work that has gone on, and there are some other risks. The used car pathway is probably one for us, and sometimes you hear Steve Gilbert, who's in charge of the border space, talk about pine needles holding things up, and so they had to come up with ways to circumvent that. Well, we're concerned about the fact that they're not stopping things when they're found, but we have to be pragmatic as well and allow trade to flow. In the domestic space, that's on that side. We've got a lot to do in terms of domestic reduction. I think the industry has probably become a little bit complacent because we haven't had any issues for a while, but it has recognised that and is making moves to try and get the foundational work done and then look at what we put on top of that to help the industry manage its own risks and extend that biosecurity system right into our forests. Readiness is probably one area where we have done pretty well. I'm running out of time very quickly, so we'll whip through these very quickly. So the industry invests a lot in surveillance. It's one of the only industries that has a national surveillance program. In fact, it has 3 programs that combine together to contribute a national surveillance system. One of those is in partnership with MPI, and then the other 2 are industry funded for industry good, and they complement a lot of the other activities that are happening, whether it's raising awareness through Find a Pest, some of MPI's initiatives and surveillance programs as well, and all of those contribute to early detection, which I'm sure you can imagine for the forestry sector that extends from the top of the North Island to the bottom of the South Island, 2 million hectares is a pretty big ask to try and do surveillance in. So we do what we can. The other part of readiness, I guess, and I've tried to signal the journey we've been on a little bit here. So when we first joined GIA in 2015, our attempts at doing readiness in partnership didn't go so well with MPI. So in some cases we went along on our own. And things have improved as we've gone on. We only have one readiness manual or contingency plan, if you like, for pitch canker. and that was a bit of an interesting journey. But more recently, there's been some really good examples of engagement, whether it's through Plant Pass, the Lepidoptera work that Matt talked about earlier as well. So all of those, though, have a common factor that they're multi-sector, so we're still struggling to get industry-specific readiness on the table with MPI, but we're still working with them and standing at the door and knocking, so we'll see what we can do. We haven't had a lot of responses, and that's probably a good thing. And again, similar journey. First few are the exact opposite of what you would have hoped to have achieved through GIA. Basically, everything that GIA promised was not achieved in those responses. And as we've gone on, in fact, we haven't had any responses since those first two. As we've gone on, we've got really good engagement and investigations. We've been able to contribute and work together with MPI and do our own risk management as well, which has proved to be really effective. And just in June, we had an investigation where we had a full, not a response, but we did cost share it, and we made a joint decision on not to do something. So that was really good. So we're getting there slowly. And in fact, we're learning a lot from other sectors that are responding all the time. So, oops, I'm out of time. 2 minutes. And then recovery is probably a bit of a blind spot. There is a lot of work happening, not specific to biosecurity, that's going to put us in good stead, whether it's industry transformation, the primary sector strategy, but there is also work going on in other areas, so stakeholders in methyl bromide reduction, which you've possibly heard of here. They're pretty I guess, well known in the Port of Tauranga space, and then Plant Pass, and we've also done some work offshore to try and manage some of those market access risks as well, on some of the Phytophthora species that could impact trade. And then research plays a big part in underpinning everything we do across those 4 hours. And look, I'm not going to go into detail because I've run out of time, but there's a lot of partnerships. I don't have them all up here because this was a very quick brain dump, but Scion are a key provider to forest biosecurity. B3 is, I guess, a foundational program for us as well and the broader system, and there's a whole bunch of players in that space that contribute to forest biosecurity research. And then the Forest Growers Research Program, that 50% of the levy funding, and they do a whole lot of work in that space. Get there. And then just looking forward, I guess some of the challenges, there are quite a few. I've just listed a couple off the top of my head here. So big ones for us, changing societal views and tolerances. Spraying Auckland's probably not going to be something that we can do quite so easily as we have in the past. Sorry, it's meant to be the previous slide, but social media, and that probably you've seen this with COVID and various other things, the spread of disinformation very quickly can undermine your freedom to operate and to do things. So that's something we have to work very closely with MPI on, and I think that's come up in the Lepidoptera discussions. And then from an industry perspective, I guess maintaining awareness, and as Mitch touched on as well, the importance of good biosecurity, but the need to address that complacency that crawls in when people have to deal with all the other issues on a day-to-day basis. And this thing that's really important, a bit like insurance, but only happens sometimes, you've got to try and keep it at the top of the list of people's thinking. So that's probably a common issue for us. And that's me. Thank you.
Thank you, Brendan. Do you want to explain this picture here to us?
I can quickly. So I think this is a gypsy moth eradication program in the US. That's the technology that they used to use.
Okay.
Back in, I think it was the 18, doesn't have the date on it.
No.
I think it was in the late 1800s or something.
Okay.
Yeah.
Was it successful?
No, it's still there.
I think, yeah, it's there in a big way.
I think it's a control program, and it's if Forestry Bill was here today, the other Dick, he would probably talk about the example and the lessons we can learn from Canada and the US's spray programs, where they're actually going out and spraying urban areas on an annual basis, and they have no issues because it's a familiar course of just what happens every day. If we try to do that here, all hell would break loose, I suspect.
Yeah, social licence.
Yep.
Yeah. So thank you for sharing your experiences to date in your role at FOA. You do have some different challenges with having a long-term crop as opposed to other sectors being able to replace plants if we need to. But forest health does have a lot of benefits, not only for your producers, but also for New Zealand Inc as well, especially around sustainability. Your readiness program is very impressive. Having that surveillance program in place for early detection is something that a lot of sectors don't have yet, and I know that it's something that we're focusing on in Kiwifruit, so I'm very envious and would like to discuss that more with you. But also talking about the gaps that you have in capability, resilience, and internal pathways that are shared across the primary industries, so something that we could probably look at working on together. So we do have some time for a couple of questions, if anyone has some. Matt.
Thanks, Brendan. That was really interesting. So you mentioned that you rely on a monoculture, a single species, but I know you've got a strong breeding program. Are you just selecting for like wood characteristics, or are you actively looking at disease tolerance as part of that breeding program as well?
I think disease tolerance for some of those, I'll call them endemic, established pathogens like Dothostroma or some of the Phytophthora is in the I guess the characteristics that they are breeding for, the traits that they're breeding for, when they are actually showing some pretty good progress in that space. But some of the offshore ones, not so much, in fact not at all at the moment, and that's something that we are starting to look at. Are there avenues that we can explore to test some of the New Zealand strains of radiata offshore to see if they're affected by the likes of Mechanisticta or various other things. So, but we've just got to figure out how to fund that and get that on the priority of our forest growers research who like the tech and getting people out of the forests and into machines. So there's a lot more investment in that space.
Cool, thank you.
Hi Brendan, nice talk. I was wondering, can you give an example, suppose where forestry has a pest or pathogens and you're kind of managing or controlling across, tens of thousands of hectares. So how do you manage specific pest using some kind of control program or management program?
So there are a couple of big ones that have high, I guess, production impact. So Dothostroma is probably one of the bigger ones. So that causes, I guess, reddening of needles and needle cast, if you like. And it doesn't kill the trees, but it certainly slows their growth. And there is a national a spraying program that at least some of the bigger companies are involved in, where they basically spray copper over the really low volumes of copper over the forest to try and manage it. But it is very much environmentally based. So if we have a hot dry summer the year before, the following year tends to have a pretty bad impact.
So when you say spray, does it mean like you just spray from the helicopters or something, or?
Yes, mainly helicopters, but planes as well. Okay. Yeah.
Brendan, just wondered if you could talk a little bit more about the value and significance of the plant pass pathway. That's because it's something that growers will hear a little bit more about in relation to our pathway plan.
Yeah, so I wasn't around when it first kicked off, but it had its origins in myrtle rust, and I guess the linkage of the spread of myrtle rust through the nursery pathway, and I think as a pathway, nurseries are well recognised as spreaders of pests and pathogens. A lot of the nurseries don't like to say that, but they are, and they've been implicated globally. So it is a significant risk pathway, I guess, that's common to all sectors. And the difference with nurseries versus I guess where you plant things out is that they connect to everything. So if an issue turns up in a nursery, it goes everywhere where those plants goes. And from a forestry perspective, that's across the country. So it's a huge issue for us and a huge risk for us. And in fact, you could possibly say some of the Phytoptheras that we do have here are probably being spread by forest nurseries, but unknowingly before we actually knew what they were. So plant pass, I guess, it's been a bit of a challenge for forestry to get it across the line. We have finally got them across the line. All those three parties I talked about have now acceded to sign the operational agreement. The next stage for us is to work out a module that allows us to adapt the plant pass core standard to be more specific and accommodating of forestry nurseries, which tend to be bare earth nurseries. So we've got a bit of work to do there, and from a forestry perspective, we also need to understand where we can manage risk and where we might need to do some more work. So getting nurseries involved in the scheme, they don't get penalised by doing things that they have no ability to actually manage cost effectively. So, but I guess Plant Pass really is about a nationwide approach to reducing risk across that pathway. And I think for us, even if we weren't a member, we would get benefits from Plant Pass through the management of pests and other sectors that might impact ours. So there's benefits across the board.
Great. Thank you, Brendan. So our next speaker is Dr. Kerry Everett, who will be coming in remotely. I hope we have the technology in place. Rebecca? Cool. Kerry is a principal scientist at Plant and Food Research and leads the epidemiology and disease management team. So using B3 funding, Kerry and her team designed specific and sensitive PCR primers to detect PSA, and these were published 6 months just before the inclusion of PSA in November 2010. So her team were the first to diagnose PSA in New Zealand using those primers. She's also an expert in apple and avocado fruit rots. And today, Kerry will give us an update on understanding biosecurity risk with compost.
So today I'm going to be talking about biosecurity risk of compost. And so first of all, why are we doing this? And it's part of Kiwifruit Vine Health's National Pathway Plan. And the aim is to mitigate the risk of spreading plant pathogens between and among kiwifruit orchards. And the number one risk for this is biological material, and of course compost is biological material. And so the types of compost that are used on kiwifruit orchards, there's 4 main types that are applied. So compost made out of plant waste, vermicast made from biosolids, vermicast from plant material, and fish compost. And all those are applied by themselves, but they're also sometimes combined, apart from fish compost, which is always, it seems, applied by itself. They can contain inorganic and organic additives, such as manure. So the components of compost, the organic components are, of course, manure. So there's 3 types, there's poultry and animal, which is cattle, and biosolids. Plant waste can come from the garden and municipal waste, which where you take your prunings to the local tips. Food waste, so kiwifruit is actually a component of compost, or some composts, and any expired products from supermarkets. Forestry also, sawdust, paper mill waste and bark. And paunch, which comes from freezing works, which is partially digested grass from ruminant animals. There's also rendered animal waste. So effectively that means it's heat treated. So it's blood and bone, and protein, which is a hydrolyzed gelatin. Then fish waste from processing and cultures. And the inorganic components are humate, which is a brown coal derivative, and gypsum. And of all those types of components in compost, these are the ones that don't, have a very low risk of spreading plant pathogens. The heat treatment gets rid of them from some of them, and others of them, unless they're present as a contaminant from the atmosphere or whatever, there won't be any plant pathogens in those components. So Mike Ormsby did a very good study on mushroom compost to import it into New Zealand, and he found 187 different species or pest disease complexes identified as biosecurity risks in imported mushroom compost. And part of our literature review, we found 50 fungi, 14 oomycetes, 12 bacteria, 15 viruses, and 4 plasmodiophoramycetes. And those are things that, funny things that affect potatoes and stuff. So there are a total of 95 different pathogens, and those are live pathogens that were found in compost. Those pathogens that are found, of which kiwifruit is a host, include Pseudomonas ringi, which has been implicated in blossom blight, and for the fungi, Botrytis cinerea, Sclerotinia sclerotiorum, Verticillium alboacrum, and Verticillium dalei, and cucumber mosaic virus, and then the oomycetes. There's 6 different species of Phytophthora that have been found in compost. So the evidence for plant pathogen spread in compost is actually not a lot of actual experiments being done, but there was one with maize head smut, and so you can see the poor maize on the side there infected. And when you feed cattle with infected maize, then the maize grown in this dung actually became infected. And anecdotally, according to Joy Tyson, one of my colleagues, the growers know that if they feed diseased onions with white rot, then the manure from that cattle will spread the disease to new fields. Mitigation. How do you get rid of plant pathogens from compost? Well, compost production, there's sort of traditional compost is made using one of these 5 methods, and these are all used for compost produced for New Zealand. So turned windrows, passively aerated static piles, forced aerated static piles, combined turned and forced aerated windrows, and in-vessel systems. And these all have a heat step. And so that heat does mitigate plant pathogens to a certain extent. But there are limitations. And so this is an in-vessel composting system. I don't know whether you can see the mouse there, but if you can't, it's the diagram on the left. And what it shows is that in a pot or a vessel, and if you put the compost in a pit, the temperatures will go up to about 60 degrees for about 10 days. For the turned windrows, what happens is that every time you turn it, the inside of the compost reaches a very high temperature, somewhere 65, 60 degrees, or even higher. But then over time it cools down, and then you turn it again, and then it goes up again. But notice that the edges of the compost piles remain cool. And so if compost is not turned properly, there are cold patches. So although heat can kill pathogens, if the composting process is not done correctly, then they won't be killed. And so for the kiwifruit pathogens, there's actually a lot of different times and temperatures that have been recommended or found to kill the kiwifruit pathogens, and there's a lot of variability, so from 15 hours to 86 days, and that's a reflection of the starting material. It actually takes longer to compost wood than it does grass or leaves. Vermicars, which is a type of compost but not technically produced the same, is actually produced at a low temperature, at less than 35, otherwise the worms will die. But the earthworm guts and grinding teeth have the same effect as thermophilic composting. However, live plant pathogens can still survive the vermicasting. And for elimination of human pathogens, which tend to be viruses and bacteria, pre- and post-thermophilic composting is recommended to make sure that they're all being killed. So the recommendations for eliminating human pathogens are in the New Zealand standards, and those recommendations are for temperatures over 55 and for durations that vary between 3 and 15 days, depending on the method that's used for the production. For plant pathogens, there are some international standards, and they actually recommend a higher temperature, which is for a windrow, it's actually shorter than for the recommendation for the human pathogens, but it's longer for the static pile and the in-vessel being 7 days as compared to 3 days. There's actually quite a lot of certification and testing of compost that happens in New Zealand, but none of these tests are for plant pathogens, and the majority of them are to assure that the compost meets organic standards. There's no testing for plant pathogens in there. As of today, there's no external checks of the New Zealand standard composting procedures are actually complied with. And Matt told me yesterday that there were some protocols for PSA, but apart from that, there are no protocols developed for eradication of plant pathogens. So the European Food Safety Authority did a assessment of compost as a risk for importing biosecurity risk pathogens, and they stated that composting cannot eradicate plant pathogens because the processes are too variable. So the substrate that you start with, the amount of turns you do, and whether when you turn the piles, all of the material reaches the lethal temperatures. That's all way too variable. And they actually recommend that plant-derived compost is not imported, and they actually ban the importation of unprocessed manure. So the conclusions are that compost does contain viable plant pathogens. They could lead to plant disease given the right conditions of humidity, rainfall, proximity and temperature. And the recommendations are that a standard for plant pathogens is prepared for compost that's applied to kiwifruit orchards. And what would be particularly important is that monitoring is conducted during the composting process to ensure that the temperature and duration is reached for killing all the plant pathogens. And good records are maintained for traceability in case something does go wrong. And also restriction of inputs. Obviously, kiwifruit material would be the highest risk of transferring kiwifruit pathogens to kiwifruit orchards, so perhaps that shouldn't be applied to kiwifruit orchards. And additional testing could also be introduced to check for plant pathogens. And of course, obtaining compost from a reputable supplier is always a good idea. So thank you, and thanks to Andrew Fraser-McKenzie and Sonia Whiteman for searching the Zespri database and initial data compilation of compost. And thank you. Are there any questions?
Thank you, Kerry. Any questions for Kerry today? I have one, Kerry. It's Leanne from KVH here. Do we know at the moment how much compost is imported into New Zealand as opposed to prepared here? Or is it all prepared here?
Well, it's all prepared here. There are some components that are imported. That tends to be things like the protamine, the humate, the gypsum. So it tends to be the organic and the rendered, because protamine is a rendered animal product, so it's effectively boiled. So there's a very low risk that will import pathogens into New Zealand.
Thanks, Kerry. So, the key messages I took away from your presentation were the compost management's a key component of the pathway plan, as you mentioned, and there's a lot of risk that potentially needs to be managed to avoid transition to our crops. So heat is the key by the sound of it, so carefully monitoring those temperatures and durations is what we need to do to make sure that there's no residual pathogens or very low. Also sourcing compost from reputable suppliers, having good traceability records and monitoring crops. So if there's anything unusual that pops up, to get in touch with KVH or MPI to follow it up. So thank you very much for your time today, Kerry. Technology issues are a challenge for me as well, so I do understand, but I hope you enjoy the rest of your day.
Okay, thanks very much.
So next up we have Monica Walter, who grew up on a German dairy mixed cropping farm and studied agricultural engineering in Germany and worked as a farm manager for a few years before coming to New Zealand. Here she completed her Masters in Agri-Science at Lincoln University, followed by working with Philip Elmore on kiwifruit botrytis and biocontrol. While working at the then Hort Research, she did her PhD at the University of Canterbury in the Environmental Science Department. She focuses on a diverse range of crops and pathogens, working on soft fruits and trunk diseases in apples and grapes, in addition to working on oomycetes in peas and berries. Monica came to Te Puke in July 2021, where she works with Kabir and his team. She looks forward to the new challenges in the Bay of Plenty and already enjoys working with growers in the industry. Monica will provide us an update on Phytophthora biodiversity and kiwifruit. Thanks, Monica.
Thank you. Now, where do I point my pointer for the slides? Pointer at the back. Oh, at the back. Okay. Thank you for that. So I need to intro why this project. Okay, so the work is done by myself, Kabir and Caitlin from Te Puke, Chamonix, Kerry and Joy Tyson up in Auckland, Ian Horn and Ari Williams in Havelock, Preeti and Pratap and Simon down at Lincoln. So these are our Phytophthora experts in PFR. So is also Preeti and Simon's team down at Lincoln. They're doing our eDNA, and Chamonix does, and up in Auckland we do the soil baiting. So samples get sent around the country, hence a big team. And I'm fairly new to Phytophthora. I'm not new to oomycetes, but I'm new to Phytophthora. So why do we do this work? In 2019, it's a CISRIP report by Woodward and Boer. They say New Zealand should be in a state of alert for Phytophthora attacks on kiwifruit and determine the risk of Phytophthora species that are present in New Zealand. And of course, as you know, with the La Mora or the kiwifruit vine decline, that's happening in Italy. A lot of oomycetes, Phytophthora and Pythium-like organisms are associated with the kiwifruit there as well, as a complex sort of syndrome. So what are oomycetes? So you can see on the graphs there, on the top left, you have a sporangia spore, so there's non-sexual, below it is a zoospore. A zoospore, you can just see the little flagellae, so they're mobile, so they can swim towards the root. It's called chemotaxis. They sense the chemistry exudated from the root tips, and this is how they find the hosts. They swim towards it. Then you have, I think it's chlamydospores up top on the right, and then the sexual stage, the oospores. That's why it's an oomycetes. They belong to the chromista, which the algae also belong to the chromista. Often the Phytophthora refer to them as straminophiles, so to differentiate from the algae. But what that indicates to you that they belong really with the algae is they're very plant-like and they love the water. So, and that is really key for Phytophthora. In terms of, but they behave a little bit like a fungus. That's why we refer to them incorrectly as fungi as well, because they're oomycetes, actually in their own kingdom. Oomycete is Greek and it translates to egg fungus with all the round bodies that you can see there. And they really cause so many diseases in so many crops. So we have the downy mildews, we have the blights, we have the dumping off, we have seed rots, we have fruit and tuber rot, we have soft rot, and penultimately we have, of course, plant death. And that's what the Phytophthora are. The word is Greek and it means plant destroyer. That's what Phytophthora means. They're really true pathogens. They're really just out to kill plants for us. And what the difficulty is about Phytophthora, and I really struggle to get my head around, hence Nadia and Ian Horner be involved in taxonomy and gender and understands it a lot much better, as well as in terms of molecular classification. In 1999, we knew around about 55 different Phytophthora species. With genetic and molecular classification, that has increased to 117, and now we're over 300 species. So not only do we get new names, but what they have been called, the names are changing. So that is very difficult in the taxonomy. And Jandin can talk here about 14 primers now, Jandin, isn't it? For classification and nomenclature. So, and that's continuously evolving, and I'm a lumper because more names doesn't help me for the biology. So taxonomy doesn't help me in terms of plant diseases and disease management. So I like to lump things. So I don't like 313 species. I'm a lot more comfortable with the 55. But, that's a trend. That's where we're sitting at. But I'm just sort of like proclaiming my own prejudices I have to the taxonomy. So the symptoms and prevention. So the symptoms, what we see in kiwifruit, and this is from Italy, a photo, and Ian Horner said, I had lovely photos, but I said you didn't send them to me, so there we go. Roots and crown show red or black diseased tissues. You have reduced terminal shoot growth, you have undersized leaves, and you have sudden or very slow vine decline, depending on the Phytophthora. Some are really rapid, and anybody who knows cauris and cauris dieback caused by Phytophthora agathidicida, we know it happens quite rapidly. And the way we combat the Phytophthora is preventing wetness and wet feet. And so we have the plants on the raised berms, on the little hills. We don't overwater, and we use the phosphorus acid-based products from control. And this is really our key for Phytophthora. It's water avoidance, and then we have our phosphorus acid products. What are we doing in the program? It's a 2-year program. It was started by Peter Scott, but he left in February. So when I inherited Peter's program, we actually restarted it. We have all of Peter's data, and we have presented that in report, but we restarted. We didn't invoice for Peter's work either, so what we have done. So it's a 2-year program. There's 4 sampling events, 2 autumn and 2 spring per year. There's 25 sites per sampling time. 4 vines per site are sampled, which gives us 100 vines per sampling event. And we sampling soil, root, and leaf analysis. The leaf analysis is to look whether we have any aerial Phytophthora. So we know that from forestry as well in apple pipfruit production, we have aerial Phytophthora, so canopy Phytophthora, but primarily it's a soil pathogen. And the targeted sampling means we're asking the growers to take us to your troubled sites. So we're looking for where you have weak vines, where you might see any Phytophthora symptoms, where you have your heavy soils, where you have your wet spots, where you have your hollows. So very targeted, so it's skewed towards finding a pathogen if it's present at all. And it is to establish diversity in kiwifruit orchards, what Phytophthora we can find. So where we have sampled, and this also includes the sites that Peter has sampled, the yellow means it's just gold variety, the green is just green, and the blue dot means is both gold and green varieties have been sampled. And I've been up in Kiri just last week, so we did Kiri. I'm going down at the end of the month to Tasman, and so we're sampling that. And we will go back to Gisborne as well in the autumn. Go. Baiting. So this is baiting, the work that Chamonix and the Aucklanders do. And as you can see, all you really do is you flood your soil, you put your different leaf substrates up top, and then within 3 days you monitor every day whether those bait substrates up top, whether they develop any water-soaked lesions, then they get picked out on a daily occasion. They get subcultured on a Phytophthora or Oomycete semi-selective agar, and then from that they get purified, and then they get sequenced. And it is a lot of work, and she can only do 6 sites per week. That's the max she can manage. Then the root samples, small root samples, kiwifruit root samples, we remove all the weeds when we sample, so we don't have weed roots in there, so we're targeting kiwifruit roots. So they get sent to Lincoln for DNA extraction from the roots, and then we have the specific PSR probes for Oomycetes and Phytophthora. And on the top gel you see all the white bands, which is, they're all positive. Every single sample has been positive for Oomycetes, but only a few of them, you see that at the bottom gel, have been positive for Phytophthora in the root samples. So what have we really found? And we really haven't found any Phytophthora species in leaf samples, so that's good news. And either in the spring 2020 or in the autumn 2021 samples using immunostrips. Admittedly, to say the immunostrips will only detect 30 different species of Phytophthora out of the 313 that we know. Low Phytophthora detection by soil baiting. From the 57 Oomycetes isolates from the last sample, from this season's autumn samples, we only found 11 as Phytophthora, and these are putatively identified, meaning we're not quite sure that these are, because we've only used ITS1 and ITS so far. It's with the COVID, the sequences are busy doing virus sequencing. So I was just waiting somewhere in the queue to be sequenced for some of the other primers. So we cannot confirm that these are indeed still what they're called, what they are, based on ITS1 and ITS4. From the eDNA, we only have 5 Phytophthora detections using the eDNA from our samples, and that's 200 root samples that have been done. Now, so I looked a little bit, or I asked Megan Shee, our knowledge navigator, to look at all the records that she can find of Phytophthora and kiwifruit. And you can see here the list, and the corresponding countries where they have been found are highlighted in black, the ones we have found, and in our kiwifruit orchards. All the Phytophthora that has been found so far, they're not new to New Zealand. And if you'll remember, sort of like the Megaspermum, the Trichicola cryptogea, they were also on the compost slide from Kerry before. So what it means, well, as a pathologist, there's really nothing to get excited about. Yeah? And I get really excited when I go out and see diseases, and the growers just look at me, with a funny face. But it's my job to get excited about pathogens. But it's really good for growers if I'm not excited. So there's really nothing to get excited about. And it's good news for the growers that I'm a little bit, okay. But what I put up there is I call it the shitty crown syndrome that I see in lots of grower properties. And I put that up because growers, when we ask growers to select us your poor vines, this is many of the places they take me to. And then you can see I already pulled it apart a little bit, either with the hunting knife or with my big spatula. And it is, I'm looking for these symptoms. The perithecia down at the bottom, the red dots, or up top, the Sporodokia. This is not Phytophthora. This is a Neonectria or Fusarium for you. That one with the perithecia, we don't know, because it could be Neonectria, could be Fusariums, could be Nectrias. Up top, that's diagnostic for Neonectria. So that's just what I wanted to put in there, because that's where they're taking me to those rootstocks, which is not Phytophthora. But I see a lot of that, and I'm sure you also have seen a lot of that. But it's a different problem. And that's me.
Thank you. Thanks, Monica. Does anyone have any questions for Monica today?
Yeah, hi. My name is Jan. Can you elaborate a little bit on that phosphorus acid influence on Phytophthora?
Oh, phosphorus acid. You might also know it as phosphonate or phosphoric acid, or you buy it maybe as Phosgard, which is sold as a fertilizer. Mind you that Phosgard sold as a fertilizer has absolutely no phosphate in it for you, and it probably takes 4 years to get any phosphate benefit out of the phosphite with I that you're applying in terms of the phosphonate. The other one that you, of course, is Elliott, which is aluminum-based phosphite. So this is very common. In the avocado growers use it for trunk injections. I think in Kauri they now use it for trunk injections with the downy mildews. I worked in boysenberry downy mildews. We use it as foliar sprays, the Phosgard. We use it for cylindrocarpin, for silverleaf control as well in apples. But in apples, when we apply it after harvest, we get residues in the fruit the following season. In kiwifruit, I don't know if you use any of the phosphorus acid products at all. Avocados used, there was 2 years of residues. So yeah, thank you. Don't do it. Yeah, it's a good question. Thank you. I'm glad we asked that, so I learned something. Whoever, just.
Yes, I managed one of the orchards was originally surveyed by Peter. I've been offered to receive a feedback about the orchard if was any Phytophthora present, and never have a news from him at all.
Chandan, would you like to answer that?
Yeah, still like 20 seconds after.
We're waiting. The letters are written. We're waiting for the permission to release them.
Do we know?
After the 22nd of November.
Do we know if any of the orchards you tested were bounty rootstock as opposed to?
We would know that. I would have to look it up, yes.
Because there is a perception that it's much more tolerant of those very wet conditions. Be interested to know whether Phytophthora has been detected or not.
But it's so little, we probably don't even pick up a rootstock trend. Yeah.
Great. I think that's all we've got time for today. Thank you very much, Monica.
Yeah, I think you should be very happy we're not finding anything.
Yeah, definitely. So thanks for your overview about the plant destroyers. I hadn't heard that interpretation before, so that was really interesting. And it's these Phytophthora are obviously an issue across the primary industries. So reassuring that you haven't found any exotic species to date, and let's hope that you continue to not be excited. Yeah. So we have one last speaker before our morning tea. So Chandan, you stand in between us and our morning tea. So Chandan Powell, who is the Innovation Manager of the Biosecurity Portfolio at Zespri, and he's been in his role since the beginning of the year. He leads the research and development activities for the biosecurity portfolio and oversees many of the projects. So prior to joining Zespri, Chandan worked as a senior scientist at Biosecurity New Zealand. So thanks, Chandan.
So I'll give you an overview of what we have done in the last 12 months in terms of the R&D work and what we're planning to do for the next 12 months. So biosecurity portfolio, we work with KBH in partnership, and our focus mainly to mitigate or reduce the pest, reduce the impact of the pest. And we normally work with, we have in our list around more than 100 different pest and disease, but nowadays our investment mostly about top 10 pest and disease. And in all those pest and disease and different level of risk. And when you think about one of the examples could be the BMSB, where it could cost for around $4.1 to $2 billion over 20 years, and the last chunk would be to Kiwifruit industry. In terms of the readiness plans, we work with KBH and we develop different set of things. And when you say readiness plan, it includes identifying, the top pest and disease for the Kiwifruit industry, understanding the impact if they come to the country, more about, what can we develop some kind of early detection techniques of the diagnostics, having a good, robust surveillance system for industry. And if they come to the country, can we set up some kind of management or eradication plan in beforehand? So all these things we do in terms of the R&D work under this portfolio. Cool. So before I just move on, I'm just to thank you, all those research providers, contractors and individuals who really work hard for us, and produce the research outcomes for our growers, and to just give the new information every other day. So there's a few R&D work that we have done over the last 12 months. And the first one is about in terms of the risk evaluation. We have done a few. So biosecurity risk of compost, Kerry nicely gave an overview, so I don't want to go back to that one. The another one for risk evaluation, we have done work on cherry leaf roll virus. There's a virus you can find in New Zealand already, and with associated a lot of kiwifruit symptoms, kiwifruit disease symptoms, including leaf spot as well as the malformation of fruits. And we try to understand the impact on goal 3. So we have done the last 12 months of work, and what we have found that yes, even when the rootstock is infected, still is a tricky virus, and you can't really find them sometimes in the scion. And we also have seen that it can retard the growth for the plant, so this is a really difficult one. Some more work is coming up for the next 12 months, and we'll see. We'll also look at the how is the impact on the subsequent fruit and the yield production as well. The next one we also dug into the Chinese literature, and Dave a little bit mentioned that one. So we have done first bit of work and second bit of work recently. So we actually uncovered a lot of new pest and disease in our list that actually was kind of unknown to our New Zealand growers as well as to us, and kind of is expanded our list. So we have more than like 50 we have found. So this new pest and disease list kind of increasing in every other day when you do the new kind of work in that space. Another piece of work we supported is a Find a Pest. Matt and a few others already mentioned that one. So it is a nice surveillance tool, and so you can do like quick identification of the pest using that tool. This Kiwifruit vine decline and trunk disease. We have focused 3 different pathogens. Ilionectria, this is one of the fungal pathogens that can actually infect a lot of wounded roots. So we have done some work in there. We also did some pathogen testing around 15 different isolates from grapevine as well. So we actually have seen that grapevine isolates can also infect the kiwifruit vines. So there should be some issues there. So that means that can be transferred somehow if there's some kind of biosecurity issues in the future. We have done some work neonectia. Neonectia is kind of emerging risk, and we heard from someone already. Monica also mentioned some of the symptoms. Growers are mentioning. So we're also looking at that one. Recently, we have done a literature review, just putting together what we have known so far in the last few years. We now know a little bit of their host range, their distribution across New Zealand in terms of South Island and North Island, Motueka, and a few other areas, a little bit more focused. There's no management options available for these fungal pathogens, but we're working on that one for the next couple of years. What are the potential management options we can also produce through our innovation work. The last one is the Phytophthora, and Monica nicely covered that one, so I'm not going to detail into that one. So next one is Ceratocystis, another fungal pathogen. The species already here in New Zealand, and the first one is reported on Kumara from 1915, and that specific species is not pathogenic to kiwifruit. So this is a good thing, though it is present here. Sometimes you can see in Kumara, but it's not really pathogenic to kiwifruit. The similar species, the same species I would say, that present in Brazil, and this has a devastating impact in the Brazil for the last 10 years. And some of our representatives from Kiwifruit and KBAs, they visited Brazil a few years back, and we have seen that what are the devastating impact that a specific fungal pathogen can do. So in terms of the readiness plan for this one, we have done some work in diagnostic space. So we produced a new molecular diagnostic assay, so we can kind of detect that pathogens early. So MPI probably going to adopt that new assay we developed as part of our R&D work. In terms of management and control, we have done some work in vitro, means work in the lab on plating. So we tested 4 different fungicides and 5 different sanitizer. Again, that specific Ceratocystis fimbriata. And what I found that the Ridomil Gold and Comet, these 2 specific fungicide actually worked better than other ones we tested, other sanitizers and fungicides. So based on those outcomes, we just move into the more like in vivo approach, means working with plants. And we tested those 2, what we have found good outcomes from our in vitro work, and we added another one as a Bion 500, another elicitor, plant elicitor. We also tested that one against 2 cultivars, the Monty from Brazil and Gold 3 from New Zealand. And what we have found that Monty was more susceptible to infection than Gold 3. Both Ridomil and Bion 500 actually reduce infection for the Monty. And for the Gold 3, New Zealand Gold 3, only the Bion 500 actually could reduce the infection. And this is some the graph you can see, the figure you can see on the right-hand side. You can see the nice, healthy plants growing. When we kind of inoculated the pathogens, when you split from the middle and inoculated, you could see some fungal infection with the black spot in the middle. When you treat it with Bion 500, we could see some of this infection kind of reduce, but still you can see on the top. So those Ridomil or Bion, neither of those fungus actually could kill the fungus, but actually just only reduce the infection level. Cool. So the next, at the BMS, we have done some work in the last 12 months. And from Zespri side, we're kind of exploring a lot of different options, and Shane will going to cover some of those, in his talk. But from the under innovation portfolio, we are focusing on the main 3 different areas in terms of the trapping, some overwintering traps, as well as some biocontrol. So some trapping, we have done some work. We developed some cheap and easy homemade traps, though those traps actually worked better early in the season, but not in the later when population was quite high. We also developed some. We actually developed some overwintering traps. So those actually try to manage the population or reduce the population size over the winter, but though we found some limited success because after the overwintering season, we could see the population level didn't really go down. So we need to do more work in that space. And the biocontrol space, we're doing some work in China, and because of the COVID, we couldn't really do in the last 12 months. So we're planning to revisit there sometime next year to do some more work in the biocontrol space. We're also keeping an eye to make sure BMSB, our Bay of Plenty is BMSB free. So we have some surveillance trap in place across Bay of Plenty. Last season, we didn't really see BMSB, which is a good thing. We also produce some training videos for our growers, so they can set up, using those training videos, they can set up the traps around the gully for the future if BMSB, just for the early detection approach. So in terms of the planning for the next 12 months, so recently we have revised our strategy, and we kind of try to do it like a more robust, future-focused strategy, and where we focus like 6 different areas. I'm just going to cover some of the projects that we're planning to do in the next 12 months. So one of the major ones we're doing like quantifying intervention. So we're trying to focus on the top 10 pests and disease and trying to just adding dollar values. So you can kind of justify which one we should be investing in. So if you suppose some of the pest has a really high impact, that means probably we need to also do a little bit more focus on those areas compared to the other ones. Though we have many in the list, but probably would be the ideal to try to do from the top, which one we should be concerned about. Next, understanding the biology space. Neonicotria is the kind of emerging one. So we are focusing more understanding the symptoms and if that neonicotria can actually transmit through bioduct. So we're focusing that one in the next 12 months. Surveillance space, we're trying to develop a new robust surveillance system, just following how the forestry has done really excellent work. So we're trying to follow that work and develop for us. Unusual symptoms, we're trying to do some work there as well. So can we kind of make it quicker, because nowadays people do unusual symptom detection. When you send to the lab, they do test for 10, 15 different tests. So can we do it in a better way? So we're trying to focus that one as well. Field diagnostics also areas we'll be keeping an eye. Management and eradication. So we are thinking about the more like excellence tracking approach. So maybe we'll be developing some kind of app or some kind of robust tracking system. So we know whether our plant products kind of moving around the country from nursery to our orchards. And if suppose there's some kind of incursion or hawkers, so we can always contain the specific nurseries or the orchards in the future, just to save the other growers from this problem. For readiness and response, readiness and preparedness plan, we're also focusing how can we better prepare the mysterious disease. For example, something we don't really know about, or some of the pest and disease is not in our list. So can we prepare ourselves for those unwanted mysterious disease a little bit early? And finally, the community engagement areas, we are focusing some work where we're trying to understand what are the social barriers or other restraint or some problems that the growers are having, so we can kind of try to lift our biosecurity practices among the growers. So with that, I'll just finish here because you're waiting for the morning tea. So thanks for your time.
Thanks, Chandon. That was a great overview of the process, projects and the research gaps that we're currently addressing. So there's a lot of interesting projects coming forward as well in the next year, and we look forward to hearing about those when we have Biosecurity Day 2022.